Hi, today we have a brief presentation on the genetic linkage of schizophrenia in the human genome. In short, many of the studies that have been conducted on this area of interest have been identifying risk loci for SNPs that may cause development of schizophrenia due to the altered expression of nervous system genes targeted by those regulatory SNPs within the loci. We hypothesize that there is a significant linkage between those SNPs and the effects of CTCF and polar 2A transcription factors that would be indicative of a causative relationship with schizophrenia. Schizophrenia, in short, is a mental illness characterized by positive, negative, and disorganized symptoms. Of these symptoms, many of them may be coinciding with environmental factors that have led to schizophrenia diagnosis. However, much of the research that's been involved with schizophrenia has been subject to focusing on genetic linkages for the disorder. By examining the risk loci associated with schizophrenia, it was found that these SNPs within the loci affected certain transcription factor binding sites that altered their binding processes and created different gene expressions for those neural regions. Schizophrenia is a mental illness characterized by the symptoms mentioned before. Many of these symptoms are cognitive effects, such as concentration and memory and focus. However, all of these symptoms are non-functional and are exhibited by patients. So, in order to identify possible functional causes for schizophrenia, many of these studies have been focused on genome-wide association in order to identify possible risk loci. And of these studies, about 180 independent schizophrenia risk loci had been identified in a study by Huo in 2019. What we ended up doing was taking the SNPs that were identified by the independent studies and comparing them in order to identify specific risk loci and how they interact with these transcription factors. In the interest of investigating the potential causal risks, these studies were conducted as a form of large-scale glosses. These 3 studies had identified 132 SNPs that had disrupted the binding sites of 21 unique transcription factors. These binding sites typically had strong chip sequence signals that were within the region, or rather the loci of interest, and these regions are typically involved with the development of neural cells and other brain cell stem lines, such that there is a possible influence of the change in expression of these transcription factors that may modify or increase the risk of the development of schizophrenia. In the comparisons where individuals with schizophrenia were investigated, about 30% of the binding sites within these regions had an increased disruption caused by the SNPs within the risk loci, and this may have led to a causal relationship between these transcription factors and schizophrenia. Among those transcription factors, polar 2A and CTCF were concentrated with the most SNPs, with about a third of the risk SNPs affecting their binding sites. An overlap was performed in order to identify which SNPs have targeted multiple transcription factors, and from those studies, polar 2A and CTCF were the most commonly bound to. Of those 97 of the 132 SNPs were significantly associated with genes in expression of human nervous system tissue, and this could be indicative of changes in the nervous system that would be causal of risks of schizophrenia. In short, many of the advancements that were made on the genetic basis of schizophrenia were focused on identifying those risk loci. However, the changes that they make in the transcription factors have a possible causal relationship to the development of schizophrenia due to there being changes in expression of the genes involving neural cells. We found that after performing a significance test, the polar 2A transcription factor was within a significant area in order to determine that the polar 2A was not bound to by random chance and does in fact have a significant effect on the relation between the risk loci affecting the transcription factors. However, CTCF, though it was the second most common, did not meet the requirements in order to verify that the results were not due to random variation. Therefore, we must reject our hypothesis, as we were only able to half confirm our findings, as there is a possible genetic linkage between polar 2A and schizophrenia. However, there is not enough conclusive evidence to dictate that CTCF and schizophrenia have a relation that would be indicative of functional changes in the genome that would result in schizophrenia. As these studies are primarily focused on SNPs, much of the functional aspects of these alterations are still unknown, and this provides a challenge for future functional genomics. Thank you, and I hope you have a wonderful day.